All right, do you hear me well? Yeah? Perfect. So thank you. So what I'm going to do now is basically present part of my PhD research, which is all about trying to understand the evolutionary relationships of the neotropical clades of vanilla. So you might be wondering why the Neotropics? Well, first of all, because it's so convenient for me, because I'm from the Neotropics, I'm from Peru. But most importantly, is because the Neotropics burst in biodiversity, representing 40% of world species and also more than 60% of plant total biodiversity. And one of the main goals of systematics is to document this biodiversity. And there is, as we heard by Alexander in his wonderful keynote, there is currently several places in the Neotropic that has important knowledge gaps, including many related to the Amazonian and also in the Andes. And although the lineages that has been used historically to understand diversity in the Neotropics usually are more concentrated in the recent highly diverse groups. What I will argue is that, and I'm not complaining by that, but what I will try to argue is that we need also to incorporate widely dispersed groups, early divergent groups, vanilla, right? So, and also because it's so much fun. It's so much fun going to the field and collect these plants. So that's another reason. That's the important reason, I think. And also, one of the most cost-effective ways of documenting this huge biodiversity has been Sanger sequencing, right? And the thing is that there are some cons and some pros about using Sanger sequencing. Being one of the benefits that is cost-effective approach, but one of the main cons is that it's, in some cases, especially to the species level, it lacks a good resolution to recover a good topology. And that's where high-throughput sequencing methods, usually combined with Sanger data, can overcome some of these limitations that give us really robust relationships, even at a species level. Just to give you an example of how we have evolved in moving from Sanger sequencing to high-throughput sequencing methods, this is one of the first phylogenetic trees of orchids published in 1999 by Kenneth Cameron, my advisor. In that work, he used RVCL, loci, and sampled close to 170 species. And just in the morning, Oscar showed us this wonderful work where he using close to 300 loci and more than 400 species, kind of give us a really interesting and kind of a broad perspective of current orchid relationships. So among the giant orchid family, the most, I will say, between the early divergent clades or first divergent clades that include Apostacioideae, Vanilloideae, and Cypripedioideae, definitely the most diverse species-rich group is Vanilla, with more than 100 species, 120 species actually. And among the giant diversity in orchids, Vanilla definitely is a rare case. It's a ruler breaker. I don't know if that's the correct term, or break ruler, right? Because if you compare this plant with all the other orchids, you will see really weird traits, like, for example, the evolution of the climbing habit, which is unique among the orchids. And also the presence of floral tubes, like in Vanilla with the fusion of the labellum with the column. And more strikingly, the presence of fleshy fruits, very light fruits that almost resemble bananas, like in this case. That has really thick, dark seeds that are really different from what you might imagine when thinking about orchids, right? So now, I hope you get the reference, but what we want to talk about is the good, the bad, and the ugly about doing vanilla systematics. And the good thing is that since we have so many Sanger data, this has given us really good clues about what are the relationships between the group. That's how we divide the genus into three major clades. Subgenus Vanilla, which is the one in green here, and is mainly neotropical. The subgenus Sanara, section Sanara, which is in orange in these figures, and is also neotropical. And this Vanilla subgenus Sanara, section Sanara, which is mainly paleotropical, with some species that migrated or dispersed to the Caribbean. And we know that the center of diversity of vanilla is the Neotropics, with about 53 species. That may change over time, but yeah, around that number. And it's highly diverse in floral morphology, as you can easily see in these images. And I'm counting this one as a good thing, despite its high taxonomic diversity and also the fact that it's one of the most known or most famous groups in orchids. We don't know much about these orchids. We don't know much about the biology, the taxonomy, the nomenclature, on the ecology. So I think that's a good thing, because that's kind of give us a fertile scene when we can do a lot of research here. So just to be clear, this is just a figure that illustrates the two major neotropical clades, subgenus Vanilla, depicted beautifully in this species, Vanilla moricensis from Peru, and Vanilla subgenus Sanara, section Sanara, which is also neotropical. And you can see that there are some differences, right? Being the main one, that subgenus Vanilla doesn't produce fragrant fruits, compared to section Sanara, we definitely produce strong fragrant fruits. And I don't know if you're surprised, but yeah, there are some vanillas that don't produce fragrance, and this is the clade that basically don't have any fragrance in their fruits. So now moving on to the bad thing is that there is no comprehensive species tree in vanilla. And that surprised me, because it's one of the most, as I mentioned before, one of the most known, one of the most famous groups of orchids. So there's no such thing as a species tree in the group. And you can definitely see that in this table that basically summarized the last decade in phylogenetic relationships in vanilla. And as you can see, hopefully in this column, the number of species and the percentage of new word species, this definitely has increased over time. And we reach a kind of a high point in about 40% for non-tropical species. But if you look to this column, which is the low side, basically we haven't moved from ITS. So that's something that, I don't know, I'm begging you, right? I'm begging you, and also, I don't know what happened, but we need definitely to move or transition from ITS to high-throughput sequencing methods. And also, phylogenetic incongruence is not uncommon in vanilla systematics. And this is due to several reasons, basically two big sets of reasons: stochastic errors, or also called technical issues, that are related to the limited character and also taxon sampling. And also there are a lot of issues about taxonomic determination that can lead to phylogenetic incongruence. But also there is another set of reasons which are called biological reasons that hasn't been explored yet, like, for example, incomplete lineage sorting, whole genome duplication, or hybridization. There's another slide about the bad thing about vanilla, which is related to monographs. So there is also the lack of an updated modern monograph in vanilla. The first one was published in 1896, and then we have another one from 1954. And then one of the most comprehensive studies about vanilla was made by Miguel Ángel Soto Arenas in 2010. It's a really seminal work, but it's hardly to consider a monograph, right? And then together with Adam and other colleagues in 2020, we published, we published this paper where we use a monographic approach to try to solve some of the names in the group. And what we are trying to do now is to transition to a modern monograph. And we are using as a kind of a model this one that was published a couple of years ago in the Ipomoea integrated, in the Ipomoea clade, or sweet potatoes. And among the two clades in the Neotropics, as I mentioned before, subgenus Vanilla, which is the one that doesn't produce any fragrance at all, and subgenus Vanilla section Sanara, which is the one that produces the fragrance, I will argue that the one that definitely needs more attention is subgenus Vanilla. Most people don't care about those vanillas because they don't produce fragrance, but I think there's a fertile kind of scene there to kind of just develop a lot of research. So among the genus, definitely this one is the most poorly understood. It's poorly sampled in the phylogenetic inference that has been done in the past decade. And many species are also poorly documented in herbaria, which makes this subgenus Vanilla a prime candidate for a comprehensive monograph of the genus Vanilla. Now, the ugly part, or I will say the super ugly part about vanilla phylogenetics is that there are severe cases of irreproducibility. And all of you here are doing science and amazing works that I've been listening for the couple of days. And you may agree with me, probably you agree with me, right? That one of the cornerstones in science is that the results must be replicable by other researchers. And that's something that is not happening in vanilla systematics. And unfortunately, it's not only on vanilla, but currently we are living in when some people are coining as a, as a reproducibility crisis. And you can see that by reading these two papers right here. I would only mention this one right here, published in 2013, that is a meta-analysis that basically sampled several phylogenetic trees and found that these studies, the phylogenetic trees in this sample, published in the last few decades, 83% of those are irreproducible due to the inability of the underlying data. That means that basically these researchers tried to replicate those trees, but they couldn't because the data was not published. So that's something that is really concerning for me, and is definitely an issue on vanilla. So now, what should we care? Why is tackling this is so important? Because having a phylogenetic tree is the starting point for asking other types of questions, other evolutionary questions. Like, for example, like, for example, once you have your tree, you're going to start asking questions about biogeography. And also, you can do some trait evolution using your tree. And definitely doing or polishing our current diversification analysis. And I'm glad that Charles mentioned this about crop wild relatives. But definitely in vanilla, being such an economically really important group, basically we are guessing who are the crop wild relatives so far, because we are lacking a strong comprehensive phylogenomic framework. So the main goals of my research is to tackle all these issues. So basically, I will try to propose a well-resolved statistical support phylogeny of the new world vanilla clades. And I will use this tree to assess the monopoly of the new world vanilla clades and resolve the relationships, hopefully, to the species level, to test the monopoly of the current infrageneric classification, to estimate when and where these neotropical lineages originate, and also propose a monograph in the subgenus Vanilla. This is part of the methods. I will not go into this section too much because I'm still learning, actually. I'm more like a traditional taxonomy, and these methods are really complicated for me at this point, but it's really exciting to be living in this era that we are witnessing these different algorithms and technology that allow us to sample entire genomes. So basically what I'm going to do is sample as many vanillas as I can and use the angiosperm 353 process to recover nuclear data sets, but also Sanger data from the NCBI websites to recover plastid loci. And then using different data sets, I will reconstruct the species coalescent tree, and also using concatenation methods, recover the biogeographic reconstruction of these clades. In terms of the monograph, what I'm going to do, and I'm basically trying to do this every day as much as I can, go into herbaria, revise as many herbaria as I can, also delve into the literature, and there are a lot of literature that we need to examine, and also doing a lot of field work, and this is really important, especially in groups that are not well known, as, for example, vanilla. And I would use these sources as a fuel for my species concept, which is basically based on my phylogenetic work, some morphometrics, phenology, and also niche modeling. So for the results, and this is just really preliminary results, something that I've made is, or that I constructed with several colleagues, is what we are, is what we are calling the Vanilla Phylogenetic Working Group. And this is really crucial because you definitely can do this type of research without involving several people, without involving different institutions, especially dealing with vanilla, which is widely distributed in the continent. So far, I have collected or assembled more than 200 specimens that represent close to 80% of the total neotropical species, with the help of 23 individuals, 23 people from different institutions, for 15 institutions and 8 different countries. There are so many of these persons here in the room, so I'm really grateful for all their help. Also, since for the past 2 years, I've been working in collecting all the data, we are still working on that, and we are planning to extracting the DNA and sequencing all the samples by the end of this year. So what I'm presenting right now is based only on Sanger data coming from 10 loci recovered from NCBI, mainly from GenBank, but also including some of my own sequences that I made in Peru, and also some of these sequences have been shared from other colleagues, like, for example, from Costa Rica, Mexico, and Brazil. So what you can see here is that at the subgenus level, subgenus Vanilla, subgenus Annata, and also the sectional level, section Thithia right here, and section Sanara right here, these are congruent with the current infragenetic classification, which is something that we expected. But if you go inner into the tree, to the group-based level, these definitely recover as paraphyletic. Also, what we did is a biogeographical reconstruction, and this is the first one that has been done since 2015 by Tom Gibnish. And there are many things to say here, but what I will highlight is that in the subgenus Vanilla, with apparently what happened is that the most recent common ancestor was widely distributed, and then at least in this clade, that I'm calling clade 1, it got restricted to the Mata Atlantica, to the Atlantic forest. And something opposite happened in the subgenus, in the section Sanara, with the most recent common ancestor being restricted to the Amazon, and then moved towards or expanded towards other areas. And in terms of the monograph, what I've been doing for the past almost 5 years is trying to document every of these species. And happily, I have been able to construct these lovely plates that not only illustrate the beauty of every species, but also give us really fundamental tools in terms of morphology. So I'm planning on recognizing 15 species so far for this subgenus. So the take-home message basically is that there is nothing vanilla about vanilla. And I don't know what means vanilla to you, but usually means plain, boring, ordinary. And it's not the case. It's really an exciting group. We need definitely urgently to transition to high-throughput sequencing methods Irreproducibility is a major problem in the group, and we need to collaborate more. Please, if you know anyone from Colombia and from Brazil, I really need people involved in this project. So I'm happy to collaborate with all of you. And finally, more related to the actual data, the infrasectional grouping in the genus is paraphyletic. And biogeographically, we are seeing some dichotomous patterns in these two neotropical groups. And in terms of the subgenus Vanilla, so far we have recognized 15 species. That's all. Thank you so much for your time.
And now we have time for 1 or 2 questions. Oh, this time it's easy. Thank you.
Thanks so much, Alexander. That was a brilliant talk, really exciting. Just a question about the monograph. Are you envisioning doing that in printed paper or some sort of hybrid e-monograph?
Sure, that's a really good question. Yeah, we have been discussing that with Ken Cameron, which is my advisor in UW Madison, and he's really he doesn't agree too much with me I'm trying to push him to, or convince him to push all the monograph online in the website of the department of the botany department of my university. That way everyone will have access to that monograph. But our plan A is to just publish it in a peer-reviewed journal, and then probably later we will kind of just upload it to the server of the university. We will see. Yeah.
Thank you very much. We still have time.
Great talk, Alexander. Such a nice project, really exciting. So I have just a quick question. If you are considering using herbarium specimens for your genomic sampling.
Yeah, that's a really, thank you, Thiago. Yeah, part of the 200 specimens that we gathered so far, the less percentage comes from herbarium specimens. Most of them come from fieldwork, which is, I think, great. But definitely there are some species that are really difficult to have samples from the field. So yeah, we're considering doing DNA extractions from the herbarium specimens. The thing is that it's quite hard to do it in vanilla. We don't know why, but yeah, it's really hard to do it. But we are trying to push in that direction, especially with Oscar, which is one of the collaborators that we have in the project.
Anyone else?
Thank you, Alexander. One of the things that always worries me with genetic analyses, and especially doing phylogenies, is that, again, often the samples are not identified by an expert and they're just sent to someone. So in these collaborative projects, just sent to someone for sequencing, and that can be highly risky, and it has been already done in vanilla in the past. So I would really, like to ask you to ask your co-authors to also prepare vouchers, at least photographs of the materials, so that you can go back and check samples, and especially in these complicated groups, it's really worth it.
Yeah, for sure. Thank you, Adam. Yeah, I'm trying to be as strict as I can with my collaborators in the fact that, yeah, we need to kind of have a really clear database for every specimens that they share, including herbarium specimens, and more importantly, also photographs of the flowers. One of the main requirements is that every specimen that enter to the project should be in flower. So we might be able to identify it. Yeah.
Great. In that case, someone in 100 years, same as Adam did, can also come back to your pictures and have another comparison. Right. Perfect. And another talk for the conference. So with this talk, we've finished for today. But worry not, tomorrow we have another day full of orchids, and on Friday also. So I'm sure that most of you will convert to orchidologists after this conference. And thank you very much.
Hopefully.
Hopefully.
Yeah.